नंतर चॅट बॉक्स मध्ये पण टाकतो. चॅट आता इमिडिएट पाठवले की शिट नेट ऑफ आहे. सॉरी पाठवले नेट ऑफ.
ಸ್ಟೋನ್ಸ್ ಕೇಳಿರಾ? ನಾಲ್ಕು ಮಂದಿ ಸ್ಟೋನ್ಸ್ ಕೇಳಿದ್ರೆ
ಕೇಳಿದ್ವಿ ಚಾಟ್ ಬಾಕ್ಸ್ मध्ये पण टाकतो. शिट नेट ऑफ आहे.
ಪ್ರಶಾಂತ್ ಬದ್ಲಿ ನಾಯಕ್ ಅಂತ ಕೊಡ್ತಾರೆ ಗೊತ್ತಾಗುತ್ತೆ. ಅವರಿಲ್ಲ ಮಂಜುನಾಥ್ ನಾಯಕ್, ಪ್ರೊಫೈಲ್ HOD ಆಫ್ ದ್ರವಗುಂಡಿ ಡಿಪಾರ್ಟ್ಮೆಂಟ್.
ಇನ್ಚಾರ್ಜ್
ಸ್ಟೇಜ್ ಮೇಲೆ ಅದ ಆನ್ ಬಿಹಾಫ್ ಆಫ್ ಪ್ರಶಾಂತ್, ಇನ್ಚಾರ್ಜ್ ಪ್ರಿನ್ಸಿಪಲ್ JR Vishnu.
So publicly they part of YouTube link. Kalan ginate. Some start.
Start ಮಾಡಕ್ಕೆ ಹೇಳ್ತೀನಿ.
Sorry.
Welcome on strength.
सर नमस्ते सर.
नमस्ते सर.
नमस्ते सर. नमस्ते.
Yes sir. Sir start.
हाँ सर. उन्होंने सर नमस्कार.
हाँ नमस्कार. बिजू कुलकर्णी, hearty welcome to the webinar. Namaste.
Thank you sir. Thank you.
Go ahead please. Good luck. Good luck. Go ahead.
Thank you sir.
Sir, I'll give you a small introduction, sir. Then we'll start, sir.
Can we start?
2 minutes, sir.
Sir, small introduction देता हूँ, मग शुरू करेगा, sir.
Introduction of dynamic personality, Dr. BG Kulkarni. Wait 2 minutes. Okay.
ठीक है. Please carry on. Go ahead.
Yes. A warm good morning to all. I, Dr. Ravi Verma, Associate Professor, Department of Sharirachna, Ayurveda Mahavidyalaya, Hubli. I welcome you all to the last session of Indriyamrutam. It gives me an immense pleasure, honor, and also I feel proud to introduce a very kind-hearted, energetic, proficient anatomist and also our college alumni, Dr. BG Kulkarni sir. Sir, we welcome you, sir. Sir is presently working as a professor and HOD in the Department of Sharirachna. Also Principal, Parul Institute of Ayurveda and Research, Vadodara, Gujarat. Sir has completed UG from Ayurveda Mahavidyalaya, Hubli. Sir has completed post-graduation from NK Japshetty Ayurvedic Medical College, Bidar. Sir has also completed his doctorate from Rajiv Gandhi University of Health Sciences, Bangalore. Sir has a teaching experience of 20 years. Sir has guided 6 PG scholars till to date. Sir is recognized PhD guide and presently guiding 8 PhD scholars under him. Sir has published more than 7 articles in various indexed and peer-reviewed journals. Sir is an executive editor of Jnanasrotas biannually journal. Currently, sir is a principal investigator in CR4D funded EMR project entitled An International Study to Assess the Augmenting Effect of Bal Chaturbhadra Arka in grade 1 and 2 undernutrition in children attending Anganwadi. With this brief introduction of sir, now I hand over the session to Dr. BG Kulkarni sir, who is supposed to speak on anatomy of skin. Sir, we welcome you, sir.
Namaskar to all. Very good morning. Thank you, Dr. Ravi. Respected Principal of Ayurveda Mahavidyalaya, Dr. Prashant A.S. sir, and all my respected teachers Now I am very happy that Dr. B.B. Hungun sir is on this platform. There are many teachers who taught me, and because of my mother institute, Ayurved Mahavidyalaya Hubli, I have reached the good position, and the blessings of all my teachers is mainly making a role in this. I remember Dr. Bhusnur Mat sir. He was a very renowned anatomist and very loving guru, I can say. Even all gurus were loving us, and we were also giving a lot of respect to all our teachers. And probably this affection and relation might have attracted us towards this webinar on this platform. Thank you very much for inviting me to share my views on this particular topic. The topic given to me is anatomy of Twacha and skin, both. And I have covered the modern description of skin and Ayurvedic description of Twacha in this presentation. I have not highlighted and not discussed Dr. Ravi.
Yes, sir. Yes, sir.
Little screen setting we are doing.
Yes, sir. Yes, sir.
Some yellow color box is coming on the screen. It is disturbing me.
Yellow color, sir.
Yep. Please move this window like that.
Switch to sir, now.
Just a minute.
Yes, sir. It was sharing again. It went, sir. Yes, sir. Yes, sir.
Okay. Okay. Thank you. I have not included the appendages of skin in my lecture because of constraint of time. I want to cover both the aspects, both Ayurveda and modern aspects.
Yes, sir.
Skin is a very important organ, and it is considered as a sense organ. There are few synonyms for this skin as a cutis and integument. Integument means the covering, which covers, which protects integument, and a separate integumentary system is considered under this heading. It is anatomically specialized boundary of a body. If you see the weight of skin, it measures around 4 to 5 kg, nearly 7% of the total body mass. And the surface area of the skin is around 1.2 to 2.2 square meter. Very huge area. And this much area is really required to maintain the homeostasis of the body, because many things in the body are depend upon the function of the skin. And the thickness of the skin varies. In a thin skin and a thick skin, the thickness varies, but 1.5 to 4 mm can be taken. And the human skin shows wide regional variations in structures like a skull, face, ear, lobule, palm, and sole, etc. This variation is as like a thickness or a number of sebaceous glands or the collagen fibers and the vascular differences, number of vessels in different parts of the skin. Okay, and because of this, by considering the role of the skin in different aspects and areas, the skin is designated as a little complex structure and highly specialized, and that is why this is referred as a sense organ. Let us go through the functions first, and then we will go to the proper anatomy. Because if we understand the function, you will understand the importance of this organ in that point of view, and how these functions are possible by the structural arrangement in the skin. Because of dermis and epidermis, most effective combination, it is most effective against microbial invasion, as the protection is the main function, and protection from the microbial is the main function. And this is very effective against dehydration and mechanical, cosmetic, thermal, photoic damage will be prevented by the skin only. And it limits and regulates the heat loss. It is a major sensory surface with elaborate system of varied receptor types. Because there are different receptors are located, they are making their role in the reception of different kinds of sensations. And it is capable of absorption with some extent and excretion also. And the skin is capable of a biochemical function like formation of vitamin D from precursor steroid under the action of ultraviolet rays. Other than this, the body heat regulation and reservoir of blood and all the other functions are also seen. Let us go to the proper structural anatomy. Microscopically, skin is divided into hairy and non-hairy or less hairy skin. These two types means thin skin and thick skin. These two types of the skin have a special peculiarities, special functions and special structures. And this whole skin is microscopically divided into three parts like a epidermis, dermis and hypodermis. The most superficial layer is dermis, below most superficial layer is epidermis. Below the epidermis, there is a layer of a dermis, which is very intimately related or connected with the epidermis, and below the dermis, there is a hypodermis. In other words, superficial fascia we call it as. Let us study what is epidermis. Actually, epidermis, what externally you are seeing is a epidermis. It is made up of keratinized stratified squamous epithelium, and this epithelium is composed of flattened interlocking polygonal cells. Okay, and mainly the epidermis is possessing four types of cells. One is the keratinocytes, another is the melanocytes, the Merkel cells, and Langerhans cells. These cells are doing their own role, and as I said, different functions, protection and all. In that point of view, these four types of cells are very important. They are doing their role. Now let us understand. Let us understand the first, keratinocytes. What is this keratinocytes? Keratin is it contains keratin, that is why it is called as the keratinocytes. And what is this keratin? Keratin is a fibrous protein. Okay, Each cell is producing a peculiar type of a protein, and so it is a fibrous protein which is making its role in the protection and protection. Okay, so in this tissue there is a continuous replacement of the cell keratinocytes. Okay, and as this move away from the base of the epidermis, undergo progressive changes. Also, changes from polygonal to dead. These are full keratinite. Hence, these are called as the keratinocytes. Means their size and shape is going to change, and they are moving from base to top. Okay, because there are different layers that I will go through in next part of the lecture. So these keratinocytes, within 25 to 40 days, the total new epidermis is going to form. And the total epidermis is predominant of these keratinocytes only. Of course, even other cells, whichever I mentioned now, Langerhans cells or the Merkel cells, they are there, but the population of this cell is very less comparing to the keratinocytes. Next cell is Langerhans cells. Langerhans cells is star-shaped. Okay, and this cell arises from the bone marrow, and from bone marrow it migrates to the epidermis, and that is why it is also called as the epidermal dendritic cell. The dendritic cells are seen usually in a CNS. Okay, but because it has got a dendrites like processes, dendrites like structure, and this cell, as it is residing in epidermis, that is why it is called as the epidermal dendritic cell. And it is a kind of a macrophage, helping to activate the immune response. Okay, because there is an invasion of the microorganisms in the skin. Many kind of debris is also going to produce. So there's a need to phagocytize these all microorganisms, and for that purpose, for protection point of view, a kind of soldiers are needed. So this Langerhans is making its role, and these macrophages are also present in dermis too. And next, very important and very conspicuous, I can say, cell is the melanocyte. If you go through the structure of a melanocyte, it is a spider-like, means it has got multiple processes, and these melanocytes are arranged in a basal layer in between the basal cells. You will understand in the next part of my lecture, and it produces the melanin granules. Okay, and these melanin pigment is packed within the small pockets, and those pockets are called, as you are seeing a small pocket in this figure, these are called as the melanosomes. And what happens, these melanocytes have spreaded their processes in a second layer. Okay, spinosum, stratum spinosum. They spread their processes in adjacent layer, and through these processes, these melanosomes will be transported. A kind of filaments are there within this processes called as the actin filaments, and along with these actin filaments, these melanosomes are transported, and melanin pigment will be taken up by the remaining cells, and this melanin is going to deposit on the superior surface of the nucleus of the cells, keratinocytes or their cells, and in this way they are protecting the layer of melanin is protecting the keratinocytes, because as you know, the important role of the melanin, it gives the protection from the ultraviolet rays from the sun and even other dangerous radiations. Okay, and speciality is this melanocyte gives the protection against the UV rays by producing a melanin and depositing it in a keratinocyte, but it is very sensitive to UV rays. Okay, so we should appreciate the function of melanocytes. And next cell is Merkel cell. Merkel cell, it is present at the junction of epidermis and dermis, and the shape is like a spiky, little small spike-like structures are there, and it is associated with disc-like sensory nerve endings. It is associated, and this disc-like nerve endings are functioning in the conduction of the sensation. Okay, so this act like Merkel disc. It is a type of receptor present in the skin, exactly at the junction of dermis and epidermis. So in carrying the sensation, this cell has got important role. Now let us discuss the layers of these keratinocytes. Okay, so this the entire epidermis is made up of the keratinocytes. As I said in the beginning, there are depending upon the thin skin and thick skin. Generally, there are 4 and 5 layers like stratum basale, stratum spinosum, stratum granulosum, and stratum corneum. This is present in a thin skin, and basale, spinosum, granulosum, lucidum, and stratum corneum. These are present in a thick skin, and so in thick skin, one extra layer of this keratinocyte comes. And these are arranged stratum basale, spinosum, granulosum, lucidum, corneum. These are arranged from bottom to top, from base to top. Okay, so our description will start from base, that is stratum basale. Stratum means strata. Strata means a layer. There is a layer of some basal cells. So these basal cells are referred as a stratum germinative also, because they are responsible for generate new cells. They are like a stem cells of the epidermis, and they divide. Mitotic division takes place. They divide, and multiple cells are going to produce. Okay, so one by one we will consider. You can refer this picture. What is this stratum basale? It is the deepest layer of the epidermis attached to underlying dermis along a wavy borderline. There is a wavy borderline. There is a nipple-like structures. The projections are there in between dermis and epidermis, and exactly at the junction of this, the stratum basale is forming a layer, single cell layer, or one layer is there. Okay, and these are involved in the cell division, because all upper the layers have to be produced by these cells only. Okay, and single row is seen. Around 20 to 25% of melanocytes and their processes are reaching the stratum spinosum. Just now I discussed this matter with you. Okay, so the more population of melanocytes. So melanocytes present in the bottom majority. Okay, and even occasional Merkel cells are also present at this junction. Next is stratum spinosum. Actually, the meaning of the spinosum, we are expecting meaning like a spine like a thorn like, but it's not like that. There are several cell layers present in this the entire thickness, and having an irregular shape. And these cells are closely packed, means they are interlocked. They are packed. Okay, and because of the desmosomes, the connecting junctions are there in between two cells. Okay, internally these cells have a number of keratin filament bundles. Keratin filament bundles, and when we do the routine histological examination, these cells are going to shrink, and because of this prominent keratin filament, it looks like having the spines on on the surface. Okay, that is why it is called a stratum spinosum. And these within these layers, particularly stratum spinosum, the Langerhans cells are more abundant. Langerhans cells. Okay, so the melanocytes and the Merkel cells majority seen in the basal layer, and Langerhans cells, as they are having a important role in defense mechanism or immunity. Okay, these are present in more population in the stratum spinosum, and the capillary supply, because the basal layer is near to the dermis, and dermis is having the blood circulation. Okay, and the capillary supply will reach up to the stratum spinosum by diffusion process. But the layers which are present above this spinosum, stratum spinosum, stratum granulosum, and all, okay, uh, it will not reach, and that is why they are prone for disintegration and even deaths also gradually. So, if we consider the stratum granulosum, there are 3 to 5 cell layers. Okay, different layers, 3 to 5, little thicker, and this shape changes drastically. There are flattened nucleus. You can see in this picture stratum granulosum. You can see there are flattened cells. The shape is changed. Previously, it was polygonal, okay, irregular, okay, and as they go up, then they change their size and shape. Okay, they become flattened, and even the nuclei also become flattened, and the organelles, other organelles present within the cell are starting degenerating. And they contain mainly 2 types of granules. That is why it is called as the stratum granulosum. This is very important. And these 2 types of granules is keratohyaline granule and laminated granules. Okay. See, this keratohyaline granule help to form a keratin in the upper layer. Okay, and the laminated granules containing a waterproofing a kind of glycoprotein, and this glycoprotein will be released in a space present beside to this all cells in the intercellular space, I can say. Okay, there these glycolipids are deposited, and that forms a type of barrier for to prevent the water loss. It is like a barrier type. Okay, and these granules are deposited. This glycolipid granules are deposited on external side of the cell membrane in this particular granulosum, and they create a resistance for the destruction because of this lipid deposition, glycolipid deposition. Okay, so because of this glycolipid, water loss can be prevented, and even water entry also can be prevented. Next is stratum lucidum. It is present only in thick skin, appear as a thin translucent band just above the stratum granulosum. A very thin band is seen, and it consists of a few rows of clear, flat, and dead keratinocytes with indistinct boundaries, because they are near to the stratum corneum, which is the dead cell layer. Okay, so they are near to that, and they have lost their boundaries, but it is full of keratin, and it is helping for the protection. Next layer is stratum corneum. Actually, stratum corneum is the thickest layer, a broad zone having 20 to 30 layers of the cells, and these are all dead cells, keratinocytes, and their plasma is very thickened because they have involved in protection from the abrasions and penetrations. Okay, and in between these cells, as I said, glycolipid, which is a waterproofing material, okay, that is going to deposit, and later these cells are going to shed off. So totally what happens, 40 to 45 days time is needed to generate a new epidermis, and these lastly the corneum, a horny cell, it is also called as a horny cell because very rough thick layer is there, and it is going to shed off, gradually shed off. Okay, and you might have seen this shedding in the form of flakes, small flakes. Okay, and we refer it as a dendrops also. They particularly scalp, in the scalp region we used to get many dead cells, dendrops. Totally it is very astonishing. Totally throughout the life, a person is shedding off around 18 kg of this cells. 18 kg. Very wonderful thing. And these cells, are dead cells, are going to shed off, and it is a good fodder for the mites. The mites present on the beds also, and mites present in your surrounding. And this the dead cell is a good fodder for that. Next layer is a dermis. We have completed epidermis here, and in epidermis we discussed the types of cells: keratinocytes, Merkel cell, Langerhans cells, and melanocytes. And the arrangement of the keratinocytes in different layers we discussed. And now let us go to dermis. See, this picture you are seeing a very beautiful picture which is showing the entire structure and the contents of this dermis. As dermis is a connective tissue, before going to talk about the dermis, I will grossly I will talk on the connective tissue. This usually, this connective tissue is like a material, connecting material, I can say in a very gross language I am telling you. And these connective tissues having a common constitution. This connective tissue is having a common constitution, like there will be a kind of matrix is present, and within this matrix the collagen fibers or different other kinds of fibers are also dispersed, and certain types of cells are also there, macrophages or mast cell or other cells, and the fibers like a collagen fibers or the elastin fibers all are present. It's just simple. You remember the wall. If you want to construct a wall, what you will do? You will put a rods and even other bars also, and then in between that there will be a framework of these all bars, and in that you put a material. Okay, within that material you may fix some material, some other electrical material or some other materials. Okay, small rods and all. So even this connective tissue also made up of this. Okay, so basically dermis is a connective tissue. Okay, and in a dermis there are two parts. One is the papillary layer and another is the reticular layer. Before going to the individual description of this, just have a look on this. Fibroblast, macrophages, mast cells, WBC, these cells are present within this dermis, in this connective tissue, and semi-fluid matrix is there. And within this matrix, the collagen fibers, the elastin fibers, and reticular fibers are embedded. Now you are getting the idea how the connective tissue is formed. Okay, so in dermis, all these material like a matrix is there, collagen fibers, elastin fibers, reticular fibers are there, and because of this constitution only, the skin is having a kind of extensibility and elasticity because of formation of collagen and elastin fibers within this material. And another thing is that it is supplied by nerve fibers and blood vessels and the lymphatic vessels. Okay, up to epidermis, up to last layer, we have seen there is a supply of a capillary to basal layer and the spinosum. Beyond that, no. Okay, but in this dermis, there is a predominant of blood vessels and lymphatics and even nerve endings, different receptors are present. And the skin appendages, okay, one of the appendage is hair, its hair root, and erector pili muscles or different sweat glands and sebaceous glands. These all are present within this layer only. They are not in epidermis. Okay, even though the root of the hair or the hair is a derivative of epidermis, but this is present in the dermis only. And the Wisner's corpuscles, Pacini corpuscle, these are all receptors, you know. These are all receptors. Even nerve endings, you can see the nerve endings in this picture, and which is surrounded to the hair root. There are some circular structures are seen in this picture. These are all Pacini's corpuscles and Wisner's corpuscles. Okay, these are all receptors. because skin is highly sensitive. You know, it is a sense organ, and it has to have a supply of these receptors, and because it is recognizing the heat, it is recognizing the touch, and even in touch also, pressure, hard touch, pain, these all sensations are received because of these all kinds of receptors present in the dermis. Yes, in this picture, you can see the arrangement of the vessels and different receptors and erector pili muscle, okay, hair root, sebaceous glands, Merkel cells, and nerve endings. So, as I said, in the dermis, there are two layers. One is the papillary layer, and another is the reticular layer. So papillary layer, the thickness comparatively is very less, and the reticular layer is thickness is more than the papillary. 1 part and 4 part, I can say, out of 5 parts. And it is a areolar connective tissue, and it is made up of collagen and elastin fibers invested with the blood vessels. And as I said, in the superior part of the papillary layer, nipple-like projections are there, and these nipple-like projections are indenting the epidermis. Epidermis. So, can you remember the corrugated cardboard? Corrugated. Okay, a wavy structure will be there, and this type of wavy structure is seen, nipple-like projection. That is why it is called as the papillary layer. Okay, and in this layer, a thick hairless skin, capillary loops, and the mucinous corpuscles are present. Just now I discussed. This layer, you will find are kind of ridges. This is very important. I want to discuss applied point of view. This is very important part to understand. You see a small lines on the skin. Okay, so basically in a palm and sole, this is seen. These dermal papillae form a dermal ridges. They form a dermal ridge. Ridge you have learned in osteology. Ridge, crest, and all such structures you have learned. Okay. So there are dermal ridges which in turn cause the overlapping epidermis to form epidermal ridges. Now, what lines you are seeing on the hand or sole, these all are epidermal ridges, but beneath that, that type of that shape, dermis is also there, and these all epidermal ridges are genetically determined, and they are very unique in each individual. Okay, even the twins are also not showing the same type of same pattern of ridges. Okay, it has got some genetic determination, and even the superficial skin is destroyed when the skin is generated there. Their pattern is not going to change. So it has got some genetic role in that. Okay, and they have got different size and shapes, I tell you, and our sweat glands are going to open in this picture, the picture which is second picture. Okay, small holes or openings are seen, and these sweat pores are going to open on these ridges, and because of this sweating and the ridge structure, we are able to produce our fingerprint or footprint. So for producing fingerprint and footprint, there is a need of a sweat also. That without that, the film will not be produced or generated. Okay, and these ridges are showing the different shapes. If you go through, it looks like a whirl, or it looks like a loop, or there are some circles, there are some semicircles or semi loops. Okay, loop is showing the right loop ridge, left loop is there, it's opened on right, opened on left, triangular ridges, circular ridges. Okay, and for kind information, listen this carefully. Why I am emphasizing these ridges? Because in certain diseases, these ridges have shown their own arrangements and even numbers also. Okay, in psoriatic cases, in a diabetes, or in a congenital blindness, or in other genetic conditions, these ridges are showing very unique arrangement, size and shape. That is why, and lot of research is going on here. The reference of these ridges is seen in Kashyapa Samhita. Kashyapa mentioned not in the modern language ridges, but Kashyapa says that there were kinds of lines present on the palm and foot sole, like a swastika shape. Even Kashyapa described the shapes also. Okay, so this is a very ancient observation. And next layer is a reticular layer, and this reticular layer forms 80% of the thickness of a dermis, and it is a dense connective tissue. And as I said, it also contains interlacing collagen fibers, and they run in various planes. They run in various planes. Okay, and most of the fibers run parallel to the skin. This is very important to understand in the dermis. These collagen fibers run in different planes. I am just emphasizing this because there are some clinical aspects are coming. I will discuss that. Most of the fibers run parallel to the skin surface, and when two lines are running parallel, in between two lines there is a sort of a gap or less dense region, and these dense regions are referred as a cleavage or tension lines. And these tension lines are externally invisible. They are not seen. Ridges are seen externally, creases are seen, or the flexures are seen. What is flexure? I will come. But these tension lines are externally not seen. Why? Because they are arranged within the reticular layer, and mainly made up of these collagen fibers. In between the collagen fibers, there is kind of a gap, and so it looks like a cleavage, And these lines run longitudinally in the skin of head and limbs, and circular around neck and trunk. Now I will show you. These are the lines you are seeing. These are lines. They are running longitudinally. They are running longitudinally. Even you can say the face and even limbs also. But they are going in a circular manner in the region of a neck and even in trunk also. In a limbs, they go in a longitudinal manner, and this has got a surgical importance and even healing process also. It is observed that when the incision, surgical incision taken across this line, tension line, or it is also called as the Langerhans lines also. Okay, and if the incision is taken against the direction, if it is running longitudinally, and in that way, if you take a circular incision, then it leads to a gaping or it forms a scar. Okay, and even healing will also be delayed. But if you take incision in the direction of these tension lines, then it helps to heal fast, and the gaping will not be there. It has got a surgical importance. And one more line you see, different creases or flexor lines, particularly on the fingers. Flexor lines are seen. Even it is seen in wrist also, fingers also, soles and toes. Okay, so these are this is the just a modification of a dermis. The dermal folds are there. These are all okay. So while learning dermis, you need to recall important receptors, then the hair follicles, hair root, different glands, such vascular arrangement, and ridges, tension line, and flexure line, because this has got applied importance. Here, almost the main part of the skin discussion I have completed, and applied aspects. Let us have the stretch mark. It is called as a stripe, particularly if you see the abdomen of a pregnant woman or a obese abdomen, kind of silvery scars are seen. It is called as a striate gravidarum in a pregnant lady, that type of marker. Why? Because there is a tearing of these collagen fibers in the present in the dermis. Dermal tearing we call it as. And in a skin cancer is another condition, as the melanin is a protective layer, or it gives a protection, but due to excess sun heat, the elastin fibers are clumped, and these UV rays can change their DNA nature, and that leads to the skin cancer. Here a few this is the picture of a skin cancer, and another is the port wine stain. Birthmarks are seen. These port wine stains are arrangement of very dense collection of dermal blood vessels on the face and neck. That is why this type of patches are seen. There is a dense collection of the blood vessels. Stretch marks you can see, white marks you are seeing. This is because of the destruction of the collagen fibers. And the tattoos, now it has got this all college people, all the models, and even players also using. I don't know what is the use of this, but the tattoos, they are giving they are pricking the needle inside the dermis, and they are pushing the pigments in the dermis, and it spreads and it remains there for a long time. That type of pigment is used there, and to remove this tattoos, they are kind of a lesser treatment is used, and so because of this lesser treatment, lesser lights, okay, these pigments molecules are going to dissolve, and these dissolved will be phagocytized by the cells, the dissolved molecules. Okay, this is, and another condition is wart. What you can see, what there is a growth of the epidermis. Okay, different layers are there. The corneum, stratum basale, different layers are seen. And one more thing is seen nowadays, piercing of the jewels. Okay, kind of jewels are pierced in the skin. They are pierced through the dermis, and they are fixed there, particularly in the nose, in the ear, even neck, even on genitals also, on eyebrows, lips, wherever they find a convenient place to show. Now let us go to the discussion of Twacha. I have 15 minutes time. So we will have a look on Ayurvedic aspects of Twacha. I purposely covered the modern aspects because if we understand the different layers of a modern aspect, you will understand the different layers as per the Ayurvedic thoughts. The Twacha is one of the Sparshanendriyam, and it is developed. Okay, Ayurveda has got its own thought of organogenesis, and it is very scientific, and they have conveyed the things by giving us similes, so that we can understand the concept very easily. Upama like Pramana. Okay, so in case of a skin, they say that tasya khalu evam pravrutasya shukra shonitasya abhiprachamanasya kshira seva santanika sapto twacha bhavanti. There are 7 layers of skin. After the fertilization of shukra and shonita, khalu eva pravrutasya shukra shonita. So after the fertilization of shukra and shonita, the 7 layers of the skin have formed. Simultaneously, all these 7 layers are not going to form. To understand this, Sushruta has given example of Kshira Seva Santanika. Means when we start heating the milk on a low flame, gradually on the surface of the milk, gradually there will be a collection of a cream, and gradually this cream becomes thicker and thicker. Okay, that is why he says Khiraseva Santanika. And if you go through the germ layers after fertilization, after morula stage, okay, first the endoderm, then the ectoderm, and then lastly the mesoderm. And from ectoderm, this the epidermis is going to form, and from the mesoderm, this dermis is going to form. So gradually, as the cream layer is going to deposit on the surface, like that, gradually there will be development. Not at a time all the layers are going to form, even in modern embryology also. So Asrubdhara, Twacha, Charma, Kali, Kuli, Sparshnendriya, Twaga, Paunendriya, these all are considered as the paryaya. And now the examination point of view, role of dosha and role of Mahabhuta in the exhibition of color to the skin. Because for the skin, there are three important factors. Even in modern also, we consider the melanin pigment and the carotene and the hemoglobin. So these are the three pigments giving the complexion to the skin. Like that, our Vata Pitta Kapha dosha and Mahabhuta are making their role in the giving the color. So by seeing the color, you can assess his Prakruti. Okay, if a Gaura Varna patient comes to you, you can say it's a Pitta Prakruti. If the Krishna Varna skin is there, there is a dominancy of Vata dosha and even Rakta also. If it is a Shama Varna, little Krishna is a dark and Sham is little quantity of the darkness come down or less. That is Vata, particularly Vata, and even Kapha and Rakta is also making the role. And Pingala is light brownish color, where the Pitta is making its role, and Rakta Dhatu. Mahabhuta, if you see the role of a Mahabhuta, Gaura Varna and Krishna Varna, two Varnas considered. And in Gaura Varna, there is a contribution of Teja, Jala, Akasha Mahabhuta. And in Krishna, there is a contribution of Teja, Parthiva, and Vayu Mahabhuta. And even in different Prakrutis, different skin texture is seen. In a Vataja Prakruti particularly, we'll see black or a blackish complexion, it will brown also. And in Pittaja Prakruti, pale yellow, reddish, whitish, yellow, moderate color yellow color is seen in a Pitta. And in Kapha, quite fair, white, and pale complexion with a luster, and very soft texture will be there. And even if you feel the skin of Vataja, Pittaja, and Kapaja Prakruti, Vataja Prakruti, you will feel it is a very dry skin, rough skin, cool and cracked skin is seen in Vataja. And in Pittaja, little warm and slightly oily and very soft skin is seen in a Pittaja Prakruti. And in a Kapaja Prakruti, very soft skin, very thick, very oily, cold and wet. Okay, and thin wrinkled skin may be seen there in a Kapaja. And if you consider skin is a Indriya, okay, so definitely it has to give a substratum to different doshas. It gives substratum to Vata dosha and Pitta dosha also, particularly the Bhajak Pitta. And skin is making important physiological role in physiology of Strotas, particularly the Mausavaha Strotas, Maus Vahedve, Tayor Mulam, Snayu Twachamcha. Snayu and Twacha, these are the two entities making the Mula for the Mausavaha Strotas. And even both Charaka and Shushruta have highlighted the Lakshanas of Twaksara Purusha. Twaksara means different types of sara are given: Twaksara, Raktasara, Mausasara, Medasara. And every sara Purusha has got a peculiar constitutions. It is called a psychosomatic constitution. Okay, even their social status also can be reflected based on these sara. So by seeing this Sahara, you can predict his health and a quality of life and quality of health. Okay, so Twak is also utilized to assess the Sahara, and that is why lot of importance has been given in the Ayurveda for Twagendriya, Twak Sahara. And best skin is Stira and Bahala Twak. Stira, very stable, wrinkles will not be there. Okay, elasticity, good kind of elasticity should be there, and extensibility is there. These all are lakshanas of Twaksara. The person who is having Twaksara is a Antas Snigdha skin, very smooth, Slakshna, Mrudu skin, and he is very pleasant, Prasanna. Okay, his a mental constitution. Sukshma, fine skin, and Alpa, normal thickness. Alpa means not taken as a less, but normal thickness. And Gambhira, Sukumara, Loma. Okay, and with luster. So these are the characteristics present in a Twaksara Purusha. And such Purusha he is very happy, having good fortune, Aishwarya power, and he enjoys these all things, and he is always intelligent and knowledgeable, and having a very healthy long life. Okay, in the Twaksara Purusha. Shushruta has described the anatomy of skin, Twacha, in a different way. He has given emphasis to the manifestation of various skin diseases in this quotation. He describes 7 layers. The first is Prathama Aubhasini, Dvitiya Lohita, Tritiya Shweta, Chaturthi Tamra, Panchami Vedini, Shashti Rohini, and Saptami Mausadhara. And even he has described the thickness of each layer also. This is very appreciable. Very appreciable. We are measuring thickness of a skin nowadays. It has become very easy. But in ancient days, they have applied their thoughts and theories, and they calculated the thickness. And this is highly appreciable. And sure, even Charak is also, but Charaka describes Shat Twacha, 6 layers, and whereas the Shushruta describes 7 layers of Twacha. Even the manner of description is same, and identification of diseases in different layers is almost same. There are some difference in the opinions, but almost the same, both the descriptions going very parallel. So for measurement of the thickness, both the Shushruta utilized the Yava Pramana, total thickness in a Yava Pramana. Okay, barley grains, you might have seen this, and they made parts of this barley grain or Yava, it is called as a Vrihi. So Shushruta Aubhasini, he says the measurement is not given, thickness is not given by Charaka. This is given by only Shushruta. So the Aubhasini thickness is 1/18 of Vrihi. If we make 18 parts of a Vrihi, 1 part of that Vrihi is to be considered as a thickness of a Aubhasini. And Lohita, 1/16th part is the Lohita thickness, and Shweta is 1/12th part of a Vrihi, and Tamra is 1/8th part of the Vrihi, Vedini 1/5th part, and Rohini is complete 1 Vrihi, and Mamsadhara is 2 Vrihi. It's a little thicker. Okay. And even Shushruta mentioned various diseases of skin. Sidma Padma Kantaka, in Avhasini, in Lohita, Tilakalaka, nature, in Shweta, that is the third layer, Charmadala, Ajagallika, Mashaka, and in fourth layer, Tamra, Kilasa Kusta, Vedini, which is the Panchama layer of Charaka, Twacha, Kusta Visarpa, Rohini, Granthi, Apachi, Arbuda, Slipada, Galaganda, and in a Mausadhara, Arsha, Vidradi, and Bhagandhara. Charaka has referred first layer as a Udakadhara because it holds the Udaka, watery part mainly. And second layer is Raktadhara, holds the blood. And then he has not nomenclated any name. He has not given any name like a Shweta, Tamra, Vedini. Just he says Tritiya, third layer, Chaturtha, Panchama, and Shat. In these layers, particularly in Tritiya layer, Charaka says Sigma Kilasa and Chaturtha Dadru Kushta. The Panchama layer, Algi Vidradi, Algae and Vidradi, and in Shat layer, there is a formation of a Arunchi having a Krishna Ratta and stool. They are stool mul in nature, means they are deeply rooted. Okay, and Mausadhara he has not mentioned. So this is the total description of Charaka and Shishruta given, very appreciable. Modern studies this in a histological way, but we study this in a disease formation and manifestation and pathology point of view. So Ovocini can be considered as a stratum corneum, horny layer, where we get sigma dermatitis. Sigma is a dermatitis. Dermatitis is a kind of inflammation of the skin. Sigma. Lohita, second layer is Lohita, Stratum Lucidum. Tilakalaka, the disease which is mentioned, such type of things are there. Vanga, and a pigmentation on the face is seen, referred as Vanga. And in a Shweta, we find Mashaka and Charmadala. So Mashaka is a mole, pigmented mole, and Charmadala kind of the surface of the skin is raised. Then Tamra, Kilasa and Dadru, such type of lesions are seen. It means that these diseases are arising from this layer, Tamra layer. Vedini is a little deeper layer where Visarpa and Kusta are seen. Visarpa, a kind of red, very burning vesicles are seen. Visarpa, very red color, highly inflamed. And Kusta, there is a destruction of the cartilages and skin. And in Rohini, you get Granthi, Galaganda. Granthi is a kind of tumors, and Galaganda, goiter-like structures, and Arbuda, cancerous growth, and Shlipada, elephantiasis or filariasis. Shlipada, very hard like a stone. And in a Mamsadhara, they say Bhagandhara, fistula in ano, and piles, external piles and internal piles, and Vidhradi is also seen. And these three diseases are arising from the Mamsadhara layer. Okay, so because of time constraint, I am unable to discuss the another few things I wanted to discuss, applied aspects. Okay, like our therapies and all, how our Panchakarma therapies are acting and all. So here I am completing my lecture. Thank you very much. And before ending, I would like to remind one anatomical thing. Few great people leave their footprint permanently, but please remember, sweat is responsible for these footprints. Just now I discussed it. Sweat is responsible for these footprints. Means for leaving footprints, there is need of a perspiration. For leaving anything, okay, there's need of a hard work. So my dear students, please do hard work and leave your valuable footprints in the field of academics. Thank you very much for giving me an opportunity to share my views with you. Thank you.
Thank you. Thank you so much, sir. Sir, we are waiting. Now the session is open for discussion. If any queries are there, please put in the chat box. Sir, there are no questions, sir.
Okay, no problem. Thank you very much, Ravi.
Yes,
And once again, I am very thankful to the organizers and particularly the principal of your college, Dr. Prashant sir, and my teacher, Dr. Hungun sir, Dr. B.B. Joshi sir, Dr. Manigol sir. There are many teachers. I cannot take names of all the teachers, but my pranam to all the teachers. Thank you very much.
yes. Thank you. Thank you.
Thank you, sir. Thank you.
Now I request Dr. Ashwin, second year PG scholar from the Department of Sharir Rachna, to deliver a vote of thanks to our resource person, Dr. B.J. Kulkarni, sir.
Ksheeraseva Santanika Sattva-Twacha Bhavanti. As the cream of the milk, 7 layers of skin are formed in the same manner. Today's resource person, Dr. B.J. Kulkarni sir, has enriched our knowledge. As the essence of topic was very well explained as the cream part of the milk. From the Department of Sharir Rachna and Ayurved Mahavidyalaya Hubli, we thank Dr. B.J. Kulkarni sir. Thank you once again, sir.
Thank you,
So, thank
Radol,
you.
and thank you, Ravi.
Thank you, sir.
Thank you so much, sir.
Okay, thank you. Please give me the feedback.
Sure, sir. Sure, sir.
Sure.
Can I leave the meeting?
Yes,
Yes,
sir.
sir.
Yes, sir.
Right.
So shortly we will move towards to our valedictory function of Indriyamrutam. Thank you, sir. Sir, ne bolo ya? Ite connect karo.
Ito connect.
कनेक्टर चाहिए 1 मिनट है